as I mentioned, we provide a large set of Jupyter notebooks that we can use in order to run the pipeline. And there's a number that we've now got available in this pipeline repository. This is the repository I was linking to from that slide. This is the JWST Pipeline Notebooks repository, where you can find all of the available notebooks that we make public right now for the different nodes that have such notebooks. All of these are designed with the idea that they're easy to get into to both process on demonstration data and also to then modify these notebooks to use them to process your own science data as well. So at the moment, this repository has notebooks for a lot of different modes: mirror imaging, slit and slitless spectroscopy, and medium resolution spectroscopy, NIRCam imaging, NIRIS imaging, and a bunch of the different NIRSpec spectroscopic modes as well. If you go to the notebooks page, then it breaks them down by each individual instrument, each individual mode, and after the session, then you can download and try out whichever notebooks you're most interested in. For today, though, then we're going to look at 3 of these notebooks that we've made available through the Science platform, just to give you an example to try running a few of these notebooks in a demo type capacity without needing to set up your own computer in order to handle them. So looks like I've now got my Science platform loaded, so if you're following along, then please go ahead and load your own Jupyter Lab as well, and you can kind of follow through what I'm doing here. As we heard earlier, the notebooks themselves are in References, Webinar Prep, and JWebinar 39. I'm going to start with running the mirror imaging notebook, which is this one here. So I've double-clicked on to open it, and I will press this kind of double right arrow here to start the kernel and run all of the different cells. And as that's running, then I'll talk through the notebook, go from top to bottom and say what it's actually doing. And it's actually going to run sufficiently fast that by the time I get to a given cell to talk about it, then it should be done. So in each one of these notebooks, then they're designed so that you can use them either for demo data or for your own science data and be highly customizable to any given set of data that you might be interested in trying to process. There's a bit of blurb at the beginning that tells you a bit about what the mode is that's processed, what kind of example data it is, but below is where we get into the really interesting stuff. The first section is the configuration section, and we've tried to design it so that this cell here should be the only cell you need to modify anything in order to process data from different programs. There's lots of cells in the notebook, we'll go through that in a moment, but this is the important cell. This is the one that you want to work with to change what data you're processing and how it's processing it. The main thing it does now here at the top is it sets whether or not we're working in demo mode. Demo mode is true by default, meaning it's going to download example data from MAST and process it through in an example way. That means that you should just be able to do what I did right now and hit go on the notebook, and it should run from end to end with example data. Once you're happy that you're good working with example data, though, and you say, no, I've got my own observing program, I want to process my data, then what you do is you set demo mode to false, and you can start configuring information about where is your data. So for instance, now you would point it here to, this is the directory that you have your science data in. So for me, I've got a flight data area on my computer linked by what the APT program number is, the observation number, and the data files live in there. If there's any background observations associated with it, you can link to those here as well. And this bottom part is where you actually set, I'll make that a little bit bigger so people can see it more easily, is where you set what parts of the pipeline you want to run. So I told you about the different pipeline stages, the detector 1, image 2, image 3, etc. Here's where you say which ones you want to do. At the moment, it's set up assuming you want to start from uncalibrated files. But if you're happy with the rate files that you get out of master already, you just want to run later stages, well, you can turn off that first stage if you wanted to, so you wouldn't run that. You can choose which ones you want to do. And you can choose what you want to do, in this case, about background subtraction. If you have a background, let's subtract it off. The details will all differ a little bit from instrument to instrument based on how they're set up. But this is the main cell that you want to modify when you go from working with demo data to working with your own data. After that, everything's optional, and we'll step through why you might want to change some of those optional things now. This next section is how it's figuring out where the reference files are. So it's setting the calibration reference data context and server. This is set up so that it looks and sees whether or not you've set all the information in your environment already. And if not, it sets up something for you so it works. You should very rarely, if ever, have to modify this section. If you want to override reference file versions, you can change the context there. You can point to a different set of reference files by uncommenting that line. I don't recommend that because different sets of reference files don't necessarily work with different pipeline versions they're not meant to work with. So buyer beware, but you can make changes there if you want to. Second section is largely uninteresting. You shouldn't ever need to modify anything. Here's where we do the standard import of packages. So import things like Astro query, import AstroPy, and the actual web pipeline itself. And that's interesting. And I just remembered the thing I forgot to do when I started this notebook, which I forget sometimes in a platform environment. You need to choose the kernel you're trying to run on. In this particular case, we need to use the master class kernel. And I will set that to the master class kernel and set things off going again. And now hopefully it should go back through that stage that it crashed at before because it couldn't find all of the things that were necessary for the notebook. Okay, and now you can see it's gone through and it's imported all of those different packages properly. It said I'm using calibration pipeline version 1.17.1. Here's the CRDS reference file context associated with it. The third section, as the title says, you can ignore if you're not using demo mode data. This is essentially just boilerplate that we added in saying if you're using demo data, what is the demo data? How do you get it from the MAST archive, etc. So this is something you don't need to configure, but if you're interested in seeing examples for how to download data programmatically from MAST via Astro query, these cells will show you how to do it. And here at the bottom, for instance, then you can see that it has downloaded all of these files that it needs for the demo. Section 4 is where things get a bit more interesting, and here's where we do the directory setup. We organize the data a little bit into different stages of the directories so that you can keep things straight. You can see where the detector 1 outputs are, the spec 2 outputs are, etc. So in that data directory linked to above, we have a uncal folder. This is where your uncalibrated data lives. You've got a stage 1 folder, the output of detector 1, similarly for stage 2 and stage 3. If you want to start changing these, you're welcome to. One of the things I often do when I'm processing data is I will make many different versions of the reductions. I'll try reducing the data 5 different ways, and I'll send each one to a different output. So if I was rerunning it, I might send it out into stage 3 underscore rotated. So if I wanted to rotate the IFU data cubes, I might send it into a different output directory, just to help keep things straight and organized. But for right now, though, you can leave it just fine with the default settings. Section 5 is where we actually get into running the pipeline. So this is the detector 1 pipeline, the thing that takes us from uncalibrated data through to ramp fitting and a given image for each individual exposure. The way this is set up is it defines a dictionary configuring how the pipeline should run. So this is a pipeline dictionary that defines each of the individual steps in the pipeline, and then has a bunch of options for changes you might want to make to those steps. So it's not an exhaustive list of options you might be able to change, it's just an example. So if you want to see the syntax for how do you make such a change, here's some examples of how you'd do it. For instance, if you wanted to make the jump step run faster by doing multiprocessing in that stage, here's the line that you'd uncomment to make the jump step run faster. If you wanted to change whether or not we were looking for cosmic ray showers in the jump step, you could uncomment that line and make changes there. There's also examples of how you would override reference files if that was something you were interested in. So this is, as I said, not complete, but it gives examples of how you'd modify things. And often there aren't going to be all that many things you want to tweak in the detector 1 pipeline. It's more going to be later stages that you're interested in. After that, it goes and finds the individual input files, both regular science files and any background files, and it will loop over those files and say, call the pipeline to run on each of these individual inputs with the set of parameters that we set up. And it will process it for all of the individual science data, and then it'll process it for the detector 1 data as well. So pipeline is running through the detector 1 stage. Once it gets to the end of detector 1, then it will go into the image 2 pipeline. And here's where we take the rate files we got out of the pipeline, and we go through doing the actual attachment of calibration information to that data. So let's just check on how the pipeline's doing up here. Yeah, there we go. So I had to restart it since I'd turned off stage 1 processing, but you can see now that it's running through. It's fetching various pieces of information from the calibration reference data server. It's going to log a bunch of information about how you've got the pipeline itself configured. And then it's going to start printing out a bunch of information about how it's going through the pipeline. Each step it runs, how the step itself actually went, and by the time that finishes, that's going to log what each output frame looks like. So while that's running, I will keep going down to the image 2 pipeline. And this is set up in the same kind of a way to what we had before. So we again set up a dictionary to describe what the individual steps in the image 2 pipeline are. And then we have some examples of ways you might want to change those pipeline settings. So for instance, if you wanted to change how you're doing the sigma clipping of the background data when you're using background subtraction, and depending on how well you did your image around, you might want to try tinkering with this. Here's an example of how you might try doing that. Likewise, if you want to turn on or off steps, here's how you would turn on or off steps here. So if you wanted to skip the resampling step, for instance, you could uncomment this line and turn it off. We also start working with associations file here, in the sense that you're now linking together multiple files. If you're doing background subtraction, you have a science file and a background file. You need to link those together. So there's a function here to define how to write those association files. Once we have that function, we can find all our science files, find all our background files, and we can loop over them and say, make an association, and then run the pipeline on that association. So again, we're calling the image 2 pipeline with the association file we made and the parameter dictionary that we set up. It's going to run through that. It's going to download any files it needs out of CRDS. It's going to tell you what all of your parameters are. And then if you want, you can scroll through the pipeline log to see what it's actually doing at any given stage. In this case, the image 2 pipeline runs really fast for mirror imaging data. So it already said that it's done, and it wrote out the results. And now there's some additional stuff in these notebooks as well. They'll sometimes tell you what steps were run, what steps were skipped, just for information purposes while you're looking through the notebook. Then the final stage of the notebook is the image 3 pipeline. Again, this is where you co-add together information from multiple different kinds of exposures. Follows the exact same structure as we had for the other parts of the pipeline. Define a Python parameter dictionary with various different steps, and give some options here about which steps you might want to configure or skip, etc. So if you wanted to change how outlier detection was happening, for instance, you might want to comment this line and change the numbers. And as I'll talk about a little bit later, then you can find out what parameters you might want to modify from our documentation on JDocs. So that'll give more information about, hey, if you've got this kind of data, we suggest you tweak this parameter. Here I'm just showing you how you would tweak those parameters. So with some examples, and you could fill in everything else sort of by context based on those examples. Likewise, because it's linking together multiple different kinds of data, you need to create an association file linking them together. Here, for instance, we're now linking together lots of different kinds of images so that you can co-add them. When we actually run the image 3 pipeline on that association, it will, as always, print a log file based on how the pipeline itself ran. And it'll print out something telling you how long it took to run this thing. In this case, this is a very fast demo example, and the image 3 step only took half a minute or something like that. And finally, at the end of each one of these notebooks, then there's a little section just allowing you to do a very quick visualization of the data, just to see, does it look like what I expect? It's not designed to be something that is particularly intensive or particularly extensive either in terms of what you're doing. It's just a quick look to see what you're looking at. So here, for instance, is the mirror imaging mosaic that we just made. So you can see that's getting a fairly nice reduction of both the LEO field and the main imaging field. You can see your bright stars. You can see a bunch of faint diffuse emission in the field as well. And you can tell, hopefully, that yes, that's the field that you expected, and roughly the data quality looks pretty good. You then want to break out into a either a different notebook or a different tool in order to actually do your science analysis. These notebooks are really designed to run the pipeline and just give you an idea what came out the other end, rather than doing extensive science analysis within the notebook. It can, however, help you do a few things. So here, for instance, there's a source finder in the pipeline. They'll try finding all of these sources. And we can say, now in this case, here's the sources that are found in, here's how bright they are, and here's what it looks like in terms of the sources that are found. So you can see each of these bright points, for instance, had a source found in the middle of them. And you could look at that table up above to see about how bright they were. Again, though, here's kind of where these notebooks end. Their purpose is not to do science analysis. Their purpose is to run the pipeline and provide you with the science data products that you need to do your own analysis. So that's the MIRI imaging pipeline notebook. I'll go through a couple others quickly just as an example, but pretty much those are the exact same kind of a format. So I'll open the NearCam imaging notebook, and I'll open the NearSpec fixed slit spectroscopy notebook as well. This time I will remember to set the kernel to masterclass before I press go. And I will start off the NearCam imaging notebook and also start off the NearSpec notebook at the same time, just so that they can both be running at the same time that I'm talking. So for the NearCam imaging notebook, this is now shifting gears a little bit from mid-infrared wavelengths to near-infrared wavelengths instead, but by and large, everything's pretty much the same. There's a few details in terms of how the notebooks differ, but everything is really very similar. As above, we start out with, this is our important cell. This is the cell that you modify in order to determine what kinds of data you're running on. At the moment, it's running with demo data, but if I wanted to run it with some kind of specialized data instead for my given science program, here's where I'd point the notebook to where to find that data and how to use it. Likewise, there's these 3 stages of the pipeline: detector 1, image 2, and image 3. Here's how you determine whether or not to turn them on or off. Again, if you wanted to start with stage 1, well, fine. If you want to start with stage 2, put it in the right directory to start with, and just skip stage 1, and you can go from there in your own processing. Again, you can change the CRDS settings if you want. Otherwise, it goes through the same kind of things: importing your different packages, downloading demo data from MAST. In this case, we've got 2 different kinds of data. We've got NearCam shortwave data and NearCam longwave data. It's downloading both. And goes from there into our directory setup. Same kind of things before. Same uncal stage 1, stage 2, and stage 3 directories by default. You're welcome to change those into anything that you're interested in. In this particular case, it'll list out the test data that we're working with. We're working with some NearCam F200W data for the shortwave, and about 4 micron data at the NearCam longwave. It'll go into the detector 1 pipeline. The exact thing as the MIRI imaging notebook. Set up a dictionary to define the individual pipeline steps, and some options here for how you might change those steps. There's a couple interesting things in here for NearCam that weren't necessarily in there for MIRI. One of those being that there's a 1/f correction here. So this is a addition that we made to the pipeline recently, allowing it to correct for 1/f striping in the data. I'll show you some examples of that in a minute. But now here is the example of how you could turn that on if you wanted to. So you can now turn it on if you uncommented this line here. Set skip to false. Regretfully, it's a little bit awkward skipping steps. Skip is the default argument to a pipeline step. So skip equals true means don't do it. Skip equals false means yes, do the step. It takes a little while to get your head around that extra negative sign in there effectively, but once you get used to it, it's not too bad. So you would turn the pipeline step on here, and you can change the parameters you might send into that step below. Right now, that detector 1 pipeline step is running, so you can see it's doing the same things before. I can scroll down in the log and see, right now it's busy working on the dark current subtraction for one of those input images. And if we keep watching, then that'll go on further. It'll go through the jump detection, it'll go through the ramp fitting, and now it's finished the ramp fitting step for one of our exposures, and it's going to move on to another one of the exposures. We'll keep scrolling down in the notebook in the meantime. Likewise, there's some notes about how to explore the data. Image2 pipeline, again, the same as the MIRI imaging directory. We've got some boilerplate dictionary setup where you define the step parameters, and a few different options of things that you can tweak about Image2. When you're done with that, it actually runs the Image2 pipeline on the data and just loops over all of the individual files. Goes from there into Image3, where it combines the data together. Again, define your setup, make any modifications to parameters you want to change. One of the ones that's particularly relevant for NeoCam is this tweak-reg step. How that works is it tries to find individual sources in each frame and adjust the astrometric solution of your data to pull those into exactly where they should be. Because there's some uncertainty in where Webb's pointing on the sky at any given time, it uses a combination of guide stars and a variety of other sources as well to try to figure out where it is in the sky. That's only as good as the catalog that we're using for it, and sometimes that can be off by a small amount, especially if it misidentified a guide star. So this step can actually be quite useful in order to improve how your NeoCam imaging steps look in terms of getting the alignments of your final mosaic looking good. So tweak-reg is something you might want to play around with in particular. Likewise, it'll create an association file from that data and then run it through the Image3 pipeline. And if I was to give it a few more minutes or so, then we'd be able to look at the output of that pipeline. And oh look, it caught up with me, great. So it ran Image3 on the shortwave data. Looks like it's running Image3 on the longwave data at the moment. So now it's doing a step called outlier detection, for instance, where it's trying to use one image to compare, does anything look weird in any images it compares it to? Are there any cosmic rays that got missed in some set of data that I can now mask out to make the data quality better? Once it's done with the outlier detection, it'll move into this resample step where it's actually trying to put all of the data together onto this common output grid so that you can co-add the data and improve the total data quality. And then it's wrapping up with the source catalog step, trying to find where are the sources in this data. Now that that's done, then it can run through various other kinds of data. This is the NeoCam shortwave data. There's both longwave and shortwave data in this notebook. So in this case, it's got two exposures it's putting together. Outlier detection is going to finish running in a moment. Then it'll go into the next stage of actually combining together the data. And then in a minute or so, we'll be done creating those data products that we can look at. There we go. It's going from outlier detection into resampling. Okay, now it's finished the resampling. It's into making the source catalog. And then it should get out into the visualization section of this. So just like the MIRI imaging notebook, there's a section at the end of the NeoCam notebook that allows you to visualize the data and see what it looks like, just as a kind of quick check to see if you're working in the right field or not. Here we're actually using a different display format. This is using some of the JWST tools that are now some of the tools provided by Space Telescope to allow users to explore the data on their own. They can be imported into notebooks, which is what we're doing here. So now here we're using it just to provide a interactive display of the data. So if we had time, I could click around through these various different options and use those to change the display, zoom into certain areas, and etc. But now there are other JWebinars on how to use this tool. Likewise, we can see, in this case, the centers of those stars look like they're saturating. There's just black holes in the middle of them. We can try overlaying the data together so we can start trying to make some pseudo-color images from the individual inputs. And just like for the MIRI imaging data, then you can visualize the source catalogs. Again, these things at the end are mostly just for example purposes, so that you can see roughly what the data looks like. Okay, NeoSpec fixed slit notebook, I'm not going to step through in the exact same kind of a way because it's the same kind of a thing. Just like with all of the pipeline notebooks for all of the modes, there's the key section up top where you configure what data you're going to try running on, the sections where you configure how it runs for each of the individual pipeline steps, and we can just skim down to the end in this case and try looking at what some of the outputs look like. This is spectroscopic data, so here you can see the dispersed trace of one of the NeoSpec fixed slits. And you can see a variety of other previews here as to what that data looks like as well. Here's the extracted spectrum that you get of it. In this case, of the 3 different nods that you observed, it's a 3-point dither. You You can see how those spectra compare to one another. Now, obviously, there are some outliers in there that'll get clipped out when you try combining together multiple different observations. And here's a final reprocessed spectrum you get out the other end. In this particular case, it looks like a nice stellar spectrum, a smoothly declining blackbody curve with a number of hydrogen-type absorption features. There's some examples at the end of this one on how you might want to modify the extraction reference file. With slit spectroscopy in particular, you don't necessarily know exactly where the source is in the slit. You might need to change up your extraction centering region slightly in order to center on your source. And there's an example in here of how you might do it. So there's now the default source here, and then a reprocessed reduction where you move that source extraction box a little bit. And you might end up with something that gives a slightly different normalization because you're now better centered on the source itself in your slit. Again, this is starting to verge on the science analysis, though, rather than pipeline. The actual pipeline steps that ran us through all of the different pipeline processing were very similar to all of the other instrument modes, and there's just some demos at the end as to what the data itself looks like and how you might want to work with it.